We will start this chapter with two words, inheritance and variation. Take an example of father, mother, and their child. Eyes of child looks like eyes of father. Nose of child looks like nose of mother. Means characters are passed on from parents to offspring. It is called inheritance of characters. In other words, it is heredity. Child also has a dimple, but both parents did not have dimple. This is called variation. Here, progeny is different from parents with respect to dimple. Humans knew about this as early as 8000 to 10,000 BC. And one more thing they know is secret of variation is hidden in sexual reproduction. They even exploited these variations. Let us see how they exploited with example. There were 100 wild horses. Among them, one horse runs very fast. Running fast is a desirable character. So they selected that horse, domesticated it, and bred this so that offsprings also get that character and run fast. This is called artificial selection and domestication. Sahiwal cows in Punjab is another example of artificial selection and domestication. Here, desirable character is high milk production. But our ancestors did not know the scientific basis for these phenomena. That is, how inheritance and variations occur over generations. The man who questioned about this phenomenon was none other than Mendel. Here, shape of the nose is character. Nose can be broad shape or narrow shape. Broad and narrow are traits. A single character may have many traits. Now, we will start to learn about Mendel's experiments from Mendel's point of view. People before Mendel know that characters or traits of a character are passed from parents to offsprings. They believed in blending theory. Scientists believed that when tall plant and dwarf plant are crossed, the height of offspring will be medium sized. According to this theory, all offsprings will be medium sized. Then think of crossing occurs between offsprings. Then what will be height of next offspring? The logical answer will be medium. Then further generations will be medium. If we apply this concept to all other characters, they also will be similar after some generations. End result should be that all individuals will be looking similar. But in reality, it is not seen. Mendel also had same questions and he wanted to find out how traits are passed from parents to offsprings with logic and not guesswork. Then he decided to do experiments on pea plants in his garden. Just to know, he studied around 28,000 pea plants and for about 7 to 8 generations. He even used other plots and lands for his study apart from his garden. In reality, Mendel studied multiple characters. For easy understanding, we will discuss single character, that is height. To start his experiments, he wanted a plant which showed consistently same character. So first, he developed true breeding lines. He developed true breeding line of tall plant and true breeding line of dwarf plant. Now, I will just give color codes to plots for easy understanding. Plots are lands where the plants are grown. Imagine now, Mendel is standing in front of blue plot. On his left side, there are tall plants, that is true breeding tall plants. Let us call it as a parent tall plant. And on his right side, there are dwarf plants, that is true breeding dwarf plant. Let us call this as a parent dwarf plant. He took pollen grains of tall plant and dusted on stigma of dwarf plant. He collected seeds from the dwarf plant. He did experiment in other way also, that is pollen grains from dwarf plant dusted on stigma of tall plants. Ultimately, we get a seeds from the offspring. Now imagine, he took 100 seeds from the offspring and planted in blue plot and waited for those seeds to grow. 100 seeds is assumed by us for easy understanding. After growth, when he saw all plants in blue plot were tall, he called them as F1 generation or filial 1 generation. So how many plants we have in blue plot? 100 tall plants. And all are similar to parent tall plant on his left side. Now, he decided to let pea plants in blue plot self-pollinate. Means pollen grains of plant are pollinating the stigma of same plant. Every plant in blue produced seeds. He collected seeds from each plant separately. Now he wants to plant seeds from each plant in separate plots to see what happens. Let us think he planted 100 seeds from each plant in yellow plots. How many yellow plots are required? 100 yellow plots. One yellow plot for each plant from F1 generation. When these seeds grown, what he observed was in each yellow plot, 75 were tall plants and 25 were dwarf. That is, 3/4 of the plants were tall and 1/4 were dwarf. He called them F2 generation. So, following observations were noted by him. The ratio is of tall to dwarf is 3:1. Tall plants were similar to parent tall plant. Dwarf plants were similar to parent dwarf plant. Dwarf disappeared in F1 generation and reappeared in F2 generation. True, no? In F1 generation, all were tall. No dwarf was seen. But again, in F2 generation, dwarf reappeared. And as previously thought, no blending is seen. That is, there were no medium sized plants. With these observations, he thought something stable has passed from parents to offsprings. He called them factors. He even observed that unchanged factor is passed from parent to offspring of F2 generation. The same factor was not expressed in F1. Now, no one tells you much about F3 generation. F2 generation gave idea about phenotypic ratio, that is 3:1. That is, plants in F2 generation were 75% tall and 25% dwarf. But next generation, that is F3 generation results are which gave genotypic ratio and some meaning to Mendel's experiment. Let us try to understand what could have happened in F3 generation. Now imagine Mendel studied further self-pollination in plants of single yellow plot. We know single yellow plot had 100 plants. 75 are tall and 25 are dwarf. Let us think next he planted seeds collected from these plants in red plots. How many plots he require? Again 100 red plots for single yellow plot. He collected 100 seeds from each plant in yellow plot and planted in red plots. When he observed after growth of plants, listen, 25 red plots showed all 100 tall plants. 50 red plots showed 75 tall plants and 25 dwarf. Another 25 red plots showed all 100 dwarf. Seeds from 25 dwarf plants of F2 generation produced all dwarf plants in F3 generation. If you see 75 tall plants of F2 generation, seeds from 25 tall plants produced all tall plants, and seeds from other 50 tall plants produced 75% tall and 25% dwarf. That is at the ratio of 3:1. Again, have you noticed? Dwarf is present in both F2 generation and F3 generation. Mendel studied further generations also, and he found dwarfs appeared later in all successive generations. So dwarfs were missing only in F1 generation. After this, Mendel applied statistics and mathematics to see passing of what number of factors from parent to offspring will give such kind of observations. First, he tried with passing of one factor and saw whether his observations fit. But they did not fit. Think, if tall factor from tall parent is passed to F1 offspring, because F1 offsprings are looking like tall parent, then self-pollination should not result in appearance of dwarf in regeneration, because when only one factor is passed, there is nothing is passing from the dwarf parent plant. So he thought dwarf parent is also passing one factor, but it's hidden in F1 and showed again in F2. Mendel realized later that for the 3:1 ratio in F2 generation to exist mathematically, the internal machinery had to work in pairs. Then he concluded that each parent carries a pair of factors. Only one of the pair is passed to the gamete, that is pollen or egg. The offspring restores the pair by taking one from each parent. And further, he told phenotype, which is expressed in F1 generation, is dominant. So here, tall is expressed in F1, so tall is dominant. The other, that is dwarf, is recessive. Tall factor is represented by capital T and dwarf factor is represented by small t. So with this, law of dominance took birth. Factors should get separated during gamete formation and again get back in offspring. This led to the birth of law of segregation. We call these Mendel's factors as genes now. Therefore, genes are units of inheritance. Genes contain information regarding expression of particular trait. If we consider capital T is gene, it has information to express tall, and small t gene has information to express dwarf. Here, tall and dwarf are contrasting traits of same character, that is height. These genes together called alleles. Alleles are slightly different form of same gene. That is, here capital T and small t are different forms of same gene. Then when to use capital T and when to use small? Capital alphabet is used for trait which is expressed in F1 generation. If you take example of other trait, among round and wrinkled, round was expressed in F1 generation. So capital R was used for round and small r was used for opposite one, that is wrinkled. When both factors are same, it is homozygous. That is, when plant has a both factors same, it is homozygous plant. When factors are different, it is heterozygous. See, in heterozygous, only one is expressed, other is suppressed. Expressed one is dominant and the other one is recessive. If you write capital T, it tells that it is homozygous for tall. If you write small t, it says that homozygous for dwarf. If you write capital T and small t, it is heterozygous for tall because tall is dominant and it is expressed. Now coming back to Mendel, he even conducted test cross to confirm his findings. Let's see the basis of test cross. He noticed that dwarfs are seen in successive generation after F2. So he considered dwarf as two recessive factors, that is, two small t's. And it was impossible to say genotype of tall plant by looking at its phenotype. So he crossed different tall plants with dwarf plant. After crossing, he observed two kinds of phenotypes. In one case, when tall is crossed with dwarf, all offsprings were tall. In other case, when tall is crossed with dwarf, 50% were tall and 50% were dwarf. In first case, genotype of tall plant should be capital T, that is homozygous. In second case, genotype of tall plant should be capital T and small t, that is heterozygous. So by seeing the number of tall and dwarf in offspring, we can tell the genotype of tall plant when it is crossed with test dwarf plant. Now let us apply the concept of test cross to the color of the flower. Before that, you should know that the recessive trait is expressed when both alleles are recessive. Dominant trait can be expressed when both alleles are dominant or even one allele is dominant, other allele is recessive. Now, take a color of the flower. If violet is dominant and white is recessive. If violet is dominant and white is recessive, plant containing white colored flowers is used for test cross. We cannot say what is the genotype of plant containing violet flowers. If violet flower containing plant is homozygous, then test cross results in all plants with violet flowers. If the plant is heterozygous, then test cross results in half of the plants with violet flowers and half of the plants with white flowers. Now let us understand about terms monohybrid cross and dihybrid cross. If you are conducting a cross, considering only one character to study, it is called monohybrid cross. Example, height. If you are conducting a cross, considering two characters to study at same time, it is called dihybrid cross. Example, yellow round seeds. Here, round is one character and yellow is second character. Remember, each character have a separate pair of genes. That is, yellow has a pair of alleles or genes, and round also has a pair of alleles or genes. Now we have seen about the monohybrid cross of the Mendel, that is tall and dwarf. Based on his observations on monohybrid crosses, Mendel proposed two general rules. To summarize his understanding of inheritance in monohybrid crosses. Today, these general rules are called the principles of laws of inheritance. First is law of dominance, and second is law of segregation. What does law of dominance say? Characters are controlled by discrete units called factors. That is, height is controlled by discrete units called factors. These factors occur in pairs. When the pair of factors are dissimilar, only one member of the pair dominates. That is called dominant factor, and the other one is recessive factor. So this law of dominance is used to explain the expression of only one of the parental characters in monohybrid cross in F1 generation and expression of both parents in F2 generation. It also explains the proportion of 3:1 obtained at the F2 generation. That is, phenotypic ratio of 3:1 obtained at F2 generation. Now, second law is based on the fact that alleles do not show any blending. Both the characters are recovered as such in the F2 generation. Though the parents contain two alleles, during gamete formation, the paired factors segregate or separate from each other such that a gamete receives only one of the two factors. Homozygous parent produces all similar gametes. Heterozygous parent produces two kinds of gametes, each having one allele with equal proportion. That is, if heterozygous parent produces 100 gametes, 50 gametes will have one factor, other 50 gametes will have other different factor. If homozygous parent produces 100 gametes, all 100 gametes will be having same factor because in homozygous both genes are same. In heterozygous both genes are different. I will tell this with respect to genes again. Homozygous parent have two genes in pairs for specific character. Both genes are same. When it produces 100 gametes, all gametes look same. On the other hand, heterozygous parent have these two genes, but both are not same. They have slight difference. So when it produces 100 gametes, 50 gametes are of one type and 50 gametes of other type. Now let us see about dihybrid crosses. Mendel also conducted dihybrid crosses, that is, inheritance of two genes. That is, two genes for different characters. With single cross, he studied behavior of two characters. If we see in sequential way how we conducted experiments, initially he crossed round seed plants and wrinkled seed plants. F1, F1 generation plants showed round seeds, so round is dominant. Same way, he crossed yellow seed plants with green seed plants. F1 generation plants got yellow seeds, so yellow is dominant. For dihybrid cross, he crossed plant with round yellow seeds and plant with green wrinkled seeds. F1 generation showed all plants with round yellow seeds. When self-pollinated, four kinds of plants with seeds formed: round yellow, round green, wrinkled yellow, wrinkled green, at ratio of 9 is to 3 is to 3 is to 1. From here, Mendel's law of independent assortment took birth. When two pairs of traits are combined in hybrid, segregation of one pair of character is independent of other pair of characters. That's why when you calculate ratio for only round to yellow separately in F2 generation, the ratio is still 3 is to 1. And when you calculate ratio for yellow and green separately, the ratio is still 3 is to 1 as seen in monohybrid cross. But when you combine both traits, then the ratio is 9 is to 3 is to 3 is to 1. To understand the possible gametes formed from parents and possible combinations they result in, Punnett square is developed. It was developed by Reginald C. Punnett. So all these possible ratios can be calculated from Punnett square. In summary, Mendel gave two laws from his monohybrid cross and one law from his dihybrid cross. Mendel conducted his experiments for 7 years, starting from 1856 to 1863. His large sample size gave credibility to data. He confirmed his findings from experiments on successive generations of his test plants. This proved that his results pointed to general rules of inheritance rather than being just unsubstantiated ideas, that is, ideas without any scientific base. Mendel conducted his experiments with 7 characters with opposing traits. So, in total, 14 traits. Means he prepared true breeding lines of 14 traits. Now, let us study the concept of dominance. What exactly is dominance? And what exactly is recessive? Imagine a gene. Genes contain information. The information is to express specific trait. Imagine gene is like a page. That page has a information. How do we write a information in page? Information in page is written with words. Same way, information in gene is written with nucleotide sequence. So, gene as a information to express a trait. In a diploid organism, means diploid organism or diploid cell, there are two copies of each gene. That is two copies of each page. These two copies are pair of alleles. If information on these two alleles or genes is identical, it is homozygous. And if information is different, it is called heterozygous. Due to some changes, information in one of the gene could have been changed. Now let's discuss with the example. Let's take an example of a gene that contains the information for producing an enzyme. Now there are two copies of this gene. Let us consider a chemical reaction in a cell where substrate S is transformed to product P. This reaction is catalyzed by enzyme we discussed. We know that gene as a information for this enzyme. Now, also let us assume the cell is heterozygous for that gene. Means information in one gene is normal and information in other gene is modified. So the normal gene has information to produce the normal enzyme. And let us see what are the possibilities with modified information on the other gene. This modified information could result in 1. Production of normal or less efficient enzyme. 2. Production of non-functional enzyme. 3. No enzyme is produced at all. If modified gene results in the production of non-functional enzyme or no production of enzyme at all, it is a recessive gene or recessive allele. And the other normal gene with normal information which can produce normal enzyme is dominant gene or dominant allele. What is incomplete dominance? Let us understand incomplete dominance. After Mendel's experiments, other scientists applied Mendel laws to study other traits in other plants. Interestingly, they found that phenotype in F1 generation did not resemble either of the parents. Phenotype was in between the two parents. Example is flower color in dog flower, also called snapdragon or Antirrhinum species. True breeding red flowered plants and true breeding white flowered plants were crossed. F1 generation showed pink flowered plants, not red or not white. Self-pollination of F1 plants resulted in development of one red, two pink colored, and one white colored. This is a phenotype ratio. If you talk in terms of seeds, like if 100 seeds from self-pollination are collected, that resulted in 25 plants with red colored flowers, 50 plants with pink colored flowers, and 25 plants with white colored flowers. So the phenotype ratio is 1 is to 2 is to 1 in F2 generation. What was the phenotype ratio in Mendel's experiment? It was 3 is to 1 in F2 generation. But genotypic ratio remained same. Why this has happened? This is because of incomplete dominance. Here, red is not completely dominant over white. Let us see how. Let's consider the gene for red is capital R and gene for white is small r. Capital R as a information to produce enzyme required for formation of red pigment. Small r as modified information so it can't produce enzyme. When plant is homozygous for capital R, that is both genes are capital R, more enzyme is produced, so more red pigment is produced, red flowers seen. When plant is homozygous for small r, no enzyme is produced, so white flowers seen. Now, when plant is heterozygous, heterozygous means one capital R and one small r. Because here only single capital R is present, less enzyme is produced, which is not enough for deep red pigment. So less pigment is produced, resulting in pink flowers. So homozygous for capital R produce red flowers. Homozygous for small r produce white flowers. And heterozygous for capital R and small r produce pink flowers. So here normal gene capital R is not completely dominant over small r. Now let us compare with the Mendel's experiment. In Mendel, in the heterozygous state, like capital T and small t, the plant will be tall. Here, in incomplete dominance, in heterozygous state, that is capital R and small r, it is not red as expected. It is pink, that is in between the two parents. Let us discuss about codominance. From Mendel experiments, we have learned that F1 resembled either of the two parents. This tells about dominance. From incomplete dominance, we have learned that F1 was in between parents. In codominance, we will see that F1 generation resembles both parents. Good example to understand codominance is ABO blood grouping. There is a gene called I. It controls blood grouping in human beings. This gene has information to produce sugar polymers. These sugar polymers are present in plasma membrane of red blood cells. They protrude from red blood cell surface. The gene I has three alleles, IA, IB, and small I. IA and IB have information which produce sugar polymers. Both sugar polymers are slightly different from each other. This is very important. Both sugar polymers are slightly different from each other. Information in small I does not produce any sugar. Because humans are diploid organisms, each person possesses any two of the three I gene alleles. IA IB are completely dominant over I. What does it mean? When IA and small I are present, only IA expresses because small I does not produce any sugar. And same way, when IB and small I are present, only IB expresses. But when IA and IB both are present together, they both express their own types of sugar. This is because of codominance. Hence, red blood cells have both A and B types of sugars. Since there are three different alleles, there are six different combinations of these three alleles that are possible. So total of six different genotypes of the human ABO blood types are possible and four phenotypes are possible. Phenotypes are A group, B group, AB group, and O group. A group and B group show complete dominance. A group, B group show complete dominance. AB group show codominance. A group phenotype have two types of genotypes. B group phenotype have two types of genotypes. AB group and O group have one type of genotype. Do you realize that the example of ABO blood grouping also provides a good example of multiple alleles? Here you can see that there are more than two, that is, three alleles controlling the same character. Humans are diploid organisms, so even single character have multiple alleles, but only two can be seen in single person. Multiple alleles of same character can be found when genes from multiple persons or population are analyzed. Mendel published his work on inheritance of characters in 1865 and it remained unrecognized till 1900, that is, for about 35 years for several reasons. First reason is communication problems. It was not easy in those days and his work could not be widely publicized. Second reason was his concept of genes or factors as stable and discrete units that control the expression of traits and pair of alleles which did not blend with each other. These two theories was not accepted by scientists at the time because continuous variation was seen in nature. Third reason was Mendel used mathematics to explain biological phenomena, which was totally new and unacceptable to many of the biologists at that time. Fourth reason was he could not provide any physical proof for the existence of factors or by what the factors are made of. So then took birth the chromosomal theory of inheritance. Then what happened in 1900? Suddenly, Mendel's work came into light. Three scientists, De Vries, Correns, and von Scherma, independently rediscovered Mendel's results. And also, advancement in microscopes allowed scientists to observe cell division. They observed doubling and division of structures inside nucleus just before cell division. These structures were observed after staining as colored bodies. They were called chromosomes. By 1902, chromosome movements during meiosis had been studied. Walter Sutton and Theodore Boveri noted that behavior of chromosomes was parallel to the behavior of genes described by Mendel. So they used chromosome movement to explain Mendel's laws. Recall that you have studied the behavior of chromosomes. The important things to remember are that chromosomes as well as genes occur in pairs. The two alleles of gene pair are located on homologous sites on homologous chromosome. If you think gene as a page, think chromosome as book. As book has many pages, chromosome has many genes. So pages appear in pairs and book also appears in pairs. Example, fifth page of one book is same as fifth page of other book in pair. That is, they are located on homologous sites. During phase 1, paired chromosomes align in midline and separated or segregated. Sutton and Boveri argued that the pairing and separation of pair of chromosomes would lead to the segregation of a pair of factors they carried. Sutton united the knowledge of chromosomal segregation with Mendelian principles and called it the Chromosomal Theory of Inheritance. I will just try to make you understand the possibilities of separation by taking two chromosomes as example. Let us think capital A and small a are pair of chromosomes and capital B and small b are other pair of chromosomes. Imagine a line and think it as a equator or midline of cell. In one scenario, capital A and capital B are above line. Small a and small b are below line. In this case, after separation, capital A and capital B go to one side and small a and small b go to one side. In another scenario, capital A and small b are arranged above line and capital B and small a are arranged below. During separation, capital A and small b move to one side and capital B and small a move to other side. So both these scenarios are possible and it's random. After that, experimental verification of the chromosomal theory of inheritance by Thomas Hunt Morgan and his colleagues led to the discovering the basis for the variation that sexual production produced. Remember, our ancestors know that secret of variation was hidden in sexual reproduction. Thomas Hunt and colleagues proved it. Morgan worked with the tiny fruit flies, Drosophila melanogaster, which were found very suitable for such studies because they could be grown on simple synthetic medium in the laboratory. They complete their life cycle in about 2 weeks. Single mating could produce a large number of progeny flies, and there was a clear differentiation of the sexes. That is, the male and female flies are easily distinguishable. And last, it has many types of hereditary variations that can be seen with the lower power microscopes. Now let us discuss about linkage and recombination. Let us understand this with simple example. Think of two books. One book is red book with 100 red pages and other book is blue book with 100 blue pages. Both books are pair of chromosomes and pages are genes. Let us understand linkage. Take red book and try to tear fifth page. Which page is likely to come with page 5? Is it page 6 or page 90? Obviously, page 6 because page 5 and page 6 are linked together. This is called linkage. That is, when two genes are near to each other, they are linked together. Next is recombination. Now swap pages from 30 to 40 page number of red and blue books. The result is red book now have blue pages from 30 to 40 page number and blue book has red pages from 30 to 40 page number. Now these books are recombinant books. Morgan carried out several dihybrid crosses in Drosophila. He studied genes that were sex-linked. The crosses were similar to the dihybrid crosses carried out by Mendel in Peace. Three concepts developed from his studies: linkage, recombination, and genetic map. For example, Morgan hybridized yellow bodied, white eyed females to brown bodied and red eyed males and also intercrossed their F1 progeny. He observed that two genes did not segregate independently of each other, and the F2 ratio deviated very significantly from the 9 is to 3 is to 3 is to 1 ratio given by Mendel. Morgan and his group knew that the genes were located on X chromosome, and they saw quickly that when the two genes in a dihybrid cross were situated on the same chromosome, the proportion of parental gene combinations were much higher than the non-parental type. That is, the gene combination which are seen in parents are seen very much higher in offsprings when compared to non-parental gene combinations. Morgan attributed this due to the physical association or linkage of the two genes and coined the term linkage to describe this physical association of genes on a chromosome. And the term recombination is used to describe the generation of non-parental gene combinations. That is, the gene combinations which are seen in offspring are not present in parents. Morgan and his group also found that even when genes were grouped on the same chromosome, some genes were very tightly linked. That is, they showed very low recombination, while others were loosely linked. They showed higher recombination. For example, he found that the genes white and yellow were very tightly linked and showed only 1.3% recombination, while white and miniature wing showed 37.2% recombination. So, when two genes are linked together strongly, they doesn't recombinate easily. When two genes are not linked strongly, then the recombination is very much possible. Next comes his student Alfred Sturtevant. He used the frequency of recombination between gene pairs on the same chromosome as a measure of the distance between genes and mapped their position on the chromosome. Like tightly linked genes, the distance between them is less. Loosely linked genes, the distance between them is more. Tight link means less recombination. Loose link means more recombination. Today, genetic maps are extensively used as a starting point in the sequencing of whole genomes, as was done in the case of Human Genome Sequencing Project. Next, we will understand polygenic inheritance and pleiotropy with simple analogy. Imagine a classroom containing kids. Each kid is a different phenotype, like one kid is color of skin, and one kid is color of hair, and one kid is height. Teacher and parents are genes. Single teacher controls all kids in school. That is, single gene controlling many phenotypes. It is called pleiotropy. When each kid reaches home, at home, mother, father, grandfather, grandmother control kid. Here, multiple genes controlling single phenotype. It is called polygenic inheritance. Let us see about polygenic inheritance. According to Mendel's studies, traits have distinct alternate forms, such as flower color, which is either purple or white. But if you look around, you will find that there are many traits which are spread across a gradient. For example, in humans, we don't just have tall or short people, but a whole range of heights are possible. Such traits are generally controlled by three or more genes. Those are called polygenic traits. Not only these traits are affected by multiple genes, they are also influenced by environment. Human skin color is another classic example for this. Here, contribution of each allele is additive. To understand this better, let us assume that three genes, capital A, capital B, capital C, control skin color. Dominant forms of capital A, capital B, capital C, responsible for dark skin color, and the recessive forms like small a, small b, small c are responsible for light skin color. The genotype with all dominant alleles, that is, two capital A's, two capital B, two capital C, will have the darkest skin color, and that with all recessive alleles will have the lightest skin color. Genotype with three dominant alleles and three recessive alleles will have an intermediate skin color. Four dominant and two recessive alleles will have color towards dark but not completely dark. Two dominant and four recessive alleles will have color towards light but not completely light. It is not just either dark or light, there is a range. Now let us see about pleiotropy. Here, single gene exhibit multiple phenotypic expression. Such a gene is called a pleiotropic gene. The underlying mechanism of pleiotropy in most cases is the effect of a gene on metabolic pathways which contribute towards different phenotypes. An example is human disease, phenylketonuria. The disease is caused by mutation in the gene that codes for the enzyme phenylalanine hydroxylase. This disease manifests as mental retardation and reduction in hair and skin pigmentation. So, here, single gene is controlling multiple phenotypes. Now, let us discuss about sex determination. Before discussing this topic, we will know what happens during gametogenesis. During gametogenesis, one diploid cell nucleus divides to give four haploid nuclei. So chromosomes gets doubled and separated. Chromosomes are two types, autosomes and sex chromosomes. Autosomes present in pairs, and let us see what happens with the sex chromosomes. The cytological observations made in a number of insects led to the development of the concept of genetic or chromosomal basis of sex determination. Henking in 1891 could trace a specific nuclear structure all through spermatogenesis, called gametogenesis, in a few insects. He observed that after spermatogenesis, 50% of the sperms received this specific nuclear structure, whereas the other 50% sperms did not receive it. Henking gave a name to this special nuclear structure as the X body. He could not explain the significance of X body. Further investigations by others fact a chromosome. It was given the name X chromosome. In large number of insects, all eggs add autosomes plus X chromosome. Some sperms add autosomes plus X chromosome. Some sperms add only autosomes. When sperm with X fertilized egg, it resulted in birth of female insect. When a sperm without X fertilized egg, it resulted in birth of male insect. So, female insect have autosomes plus XX, that is 2X chromosomes. Male insect have autosomes plus X, that is single X chromosome. So, it's called X0 type of sex determination. So, number of chromosomes in females is more because female has extra X chromosome. Grasshopper is an example of X0 type of sex determination. Again, in simple words, male have single X and female have 2X. In other insects and mammals, including human being, male have single X and single Y, and female have 2X. It is XY type of sex determination. And number of chromosomes same in both male and female. Who is heterozygous here? Male is heterozygous with one X and one Y. So it is male heterogeneity. But see, in some birds, male have two Z chromosomes and female have one Z and one W. Here also male and female have same number of chromosomes. So it is female heterogeneity. So three mechanisms of sex determination we have seen. X0 type, XY type where male heterogeneity is seen, ZW type where female heterogeneity is seen. So sex determining mechanism in case of humans is XY type. There are 46 chromosomes or 23 pairs of chromosomes. Out of 23 pairs of chromosomes present, 22 pairs are exactly same in both males and females. These are the autosomes. A pair of X chromosomes are present in female. One X and one Y is present in male. In males, during spermatogenesis, two types of gametes are produced. 50% carry the X chromosome and the rest 50% as Y chromosome. Females, however, produce, produce only one type of ovum with an X chromosome. There is an equal probability of fertilization of the ovum with the sperm carrying either X or Y chromosome. If sperm with X fertilizes ovum, it results in female child. If sperm with Y fertilizes ovum, it results in male child. It is the genetic makeup of the sperm that determines the sex of the child. In each pregnancy, there is always 50% probability of either a male or a female child. But in our society, women are blamed for giving birth to female children. Let us see about sex determination in honeybees. It is very interesting. Let's start with eggs. This egg has a 16 chromosomes. If this egg is fertilized by sperm having 16 chromosomes, zygote with 32 chromosomes form, which develop into female bee, which is diploid. It may be either queen or worker. If egg doesn't get fertilized, it develops into male bee by parthenogenesis. Here male is haploid. Again, female bee produce eggs by meiosis and male bee produce sperms by mitosis. It is called haplodiploid sex determination. Now let us see about next concept, mutation. Now we know that information in gene is present as nucleotide sequence. We can call it as a DNA sequence. Mutation is a phenomenon which results in alteration of DNA sequences, which in turn results in change in genotype, which in turn result in change in phenotype. You have seen that previously. Recombination results in variation. Not only recombination, mutation is another phenomenon that leads to variation in DNA. Mutations can be insertion or deletion of a segments of DNA or insertion or deletion of single base pair, which leads to frameshift mutation. What causes mutation? Chemical factors, physical factors. Factors which cause mutation are called mutagens. Mutation is nothing but a change in information in a gene. Let us discuss about pedigree analysis. Mendel performed control crosses in pea plants to analyze inheritance patterns. Control crosses is not possible in human beings. Alternative is study of the family history about inheritance of a particular trait. Such an analysis of traits in a several of generations of a family is called the pedigree analysis. In the pedigree analysis, the inheritance of a particular trait is represented in the family tree over generations. In human genetics, pedigree study provides a strong tool which is utilized to trace the inheritance of a specific trait, abnormality, or disease. Some standard symbols are used for pedigree analysis to know inheritance patterns at a glance. By now, we understood that every character in our body is controlled by genes. Genes are present in DNA. Most of these are passed from parents to offsprings unchanged. Sometimes mutation changes genes or DNA, and those genes may cause disease. Those diseases are called genetic disorders. Now let us discuss about next topic, genetic disorders. By now we understood that every character in our body is controlled by genes. Genes are present in DNA. Most of these are passed from parents to offspring unchanged. Sometimes mutation changes genes or DNA, and those genes may cause disease. Those diseases are called genetic disorders. Genetic disorders may be grouped into two categories: Mendelian disorders and chromosomal disorders. Mendelian disorders are due to alteration or mutation in single gene. Those disorders are transmitted to the offspring, same as Mendelian laws. Pedigree analysis can be used here to trace disorder in family. Most common and prevalent Mendelian disorders are hemophilia, cystic fibrosis, sickle cell anemia, color blindness, phenylketonuria, thalassemia, etc. Mendelian disorders may be dominant or recessive. By pedigree analysis, one can easily understand whether the trait in question is dominant or recessive. Similarly, the trait may also be linked to the sex chromosome, as in case of hemophilia. Let us see about color blindness. It is a sex-linked recessive disorder. There is a defect in either red or green cones of eye, resulting in failure to discriminate between red and green color. This defect is due to mutation in certain genes present in the X chromosome. It occurs in about 8% of males and only about 0.4% of females. This is because the genes that lead to red-green color blindness are on X chromosome. Males have only one X chromosome and females have two X chromosomes. The son of a woman who carries the gene has a 50% chance of being colorblind. The mother is not herself colorblind because the gene is recessive. That means its effect is suppressed by her matching dominant normal gene, that is, other dominant gene present on other X chromosome. A daughter will not normally be colorblind unless her mother is a carrier and her father is also colorblind. Next is hemophilia. It is also sex-linked recessive disease. Our blood clots after injury. Many proteins are involved in this process. In this disease, single protein is affected, resulting in non-stop bleeding. Again, the heterozygous female, that is carrier, for hemophilia may transmit the disease to sons. The possibility of a female becoming a hemophilic is extremely rare because mother of such a female has to be at least carrier and father should be hemophilic. It is not possible because sons with hemophilia are unviable in the later stages of life. Though that means they don't live for many years. The family pedigree of Queen Victoria shows a number of hemophilic descendants as she was a carrier of the disease. Next is sickle cell anemia. This is an autosomal linked recessive trait. Hemophilia and color blindness were sex linked recessive. Sickle cell anemia is a autosome linked recessive trait. Normally, there are two genes, hemoglobin A, hemoglobin A. Carrier will have hemoglobin A and hemoglobin S. Hemoglobin S is a defective gene. Diseased person will have both hemoglobin S. It can be transmitted from parents to the offspring when both the parents are carrier for the gene or heterozygous for the gene. They are called sickle cell trait. It affects hemoglobin molecule. Hemoglobin molecule is made up of 2 alpha and 2 beta chains. Normal beta globin chain has a glutamic acid at 6th position. It is replaced by valine. Here we see change of normal 6th codon GAG to GUG. The mutant hemoglobin molecule undergoes polymerization under low oxygen tension, causing the change in the shape of the RBC from biconcave disc to elongated sickle cell structure. This will block the blood vessels and results in symptoms. Next disease is phenylketonuria. This inborn error of metabolism is also inherited as the autosomal recessive trait like sickle cell anemia. The affected individual lacks an enzyme that converts the amino acid phenylalanine into tyrosine. As a result of this, phenylalanine is accumulated and converted into phenylpyruvic acid and other derivatives. Accumulation of these in brain results in mental retardation. These are also excreted through urine because of its poor absorption by kidney. Next disease is thalassemia. This is also an autosome linked recessive blood disease. It transmitted from parents to offspring when both the parents are unaffected carrier of the gene, that is heterozygous. Here, due to mutation or deletion of gene, it results in reduced rate of synthesis of one of the globin chains. It can be either alpha chain or beta chain. This causes the formation of abnormal hemoglobin molecules resulting into anemia, which is characteristic of this disease. Thalassemia can be classified according to which chain of the hemoglobin molecule is affected. Alpha thalassemia is controlled by two closely linked genes, hemoglobin A1 and hemoglobin A2 on chromosome 16. So total 4 genes control alpha thalassemia. The more genes affected, the less alpha globin molecules produced. While beta thalassemia is controlled by a single gene, that is hemoglobin B, which is present on chromosome 11. So total 2 genes control it because genes occur in pair. Thalassemia is a quantitative problem. Sickle cell anemia is a qualitative problem. Next, we will see about the chromosomal disorders. They are caused due to absence, excess, or abnormal arrangement of one or more chromosomes. Failure of segregation of chromatids during cell division cycle results in the gain or loss of a chromosome called aneuploidy. For example, Down syndrome results in the gain of extra copy of chromosome 21. So, Down syndrome is a type of aneuploidy. Similarly, Turner syndrome results due to loss of an X chromosome in human females. So, Turner syndrome is also an example of aneuploidy. Failure of cytokinesis after telophase stage of cell division results in an increase in a whole set of chromosomes in an organism, and this phenomenon is known as polyploidy. This condition is often seen in plants. The total number of chromosomes in a normal human cell is 46, that is 23 pairs. Out of these, 22 pairs are autosomes and one pair of chromosomes are sex chromosomes. Sometimes, though rarely, either an additional copy of chromosome may be included in individual, that is trisomy, or an individual may lack one of any one pair of chromosomes, that is monosome. Let's talk about Down syndrome. The cause of this genetic disorder is presence of an additional copy of the chromosome number 21, that is trisomy 21. This disorder was first described by Langdon Down in 1866. The affected individual is short statured with small round head, furrowed tongue, and partially open mouth. Palm is broad with characteristic palm crease. Physical, psychomotor, and mental development is retarded. Klinefelter syndrome is the next syndrome we will discuss. This genetic disorder is also caused due to the presence of an additional copy of X chromosome, resulting into a karyotype of 47XXY. Next, we will discuss about Klinefelter's syndrome. This genetic disorder is also caused due to the presence of an additional copy of X chromosome, resulting into a karyotype of 47XXY. Such an individual has overall masculine development. However, the feminine development, that is development of breast, also called gynecomastia, is also expressed. Such individuals are sterile. Next, we will talk about Turner syndrome. Such a disorder is caused due to the absence of one of the X chromosomes, that is 45 with X0. Such females are sterile as ovaries are rudimentary besides other features including lack of other secondary sexual characters.